Hey everyone, I have Anush and Mikael here today, and we are going to be discussing our new technology that we've been researching, just developing for a hackathon, and it's called USPION, which is type of nanoparticle assisted deep brain stimulation. So, Mikael, why don't you start us off and talk a little bit about what is deep brain stimulation and how is it being carried out today?
All right. So basically, deep brain stimulation basically involves implanting electrodes within certain areas of your brain. And then basically, these electrodes produce electrical impulses that regulate abnormal impulses, or the electrical impulses can affect certain cells and chemicals within the brain. As of right now, deep brain stimulation is invasive, and it requires putting a huge rod in your brain, in layman terms, which is like which causes an extreme amount of side effects, all to treat a number of conditions such as dystonia, epilepsy, um, Parkinson's, etc., etc.
Awesome. Anush, do you have anything to add?
Yeah, I mean, I would say that it's right now more of like a two-in-one package. Even if it's invasive, it definitely has its upsides in terms of that it can regulate the abnormal signals and also impulse in a timely fashion, right? Let's say it's a more of a periodic thing, then you could easily like program this DBS to like do the impulses at a certain time frequency, or you could do it to where like the EEG chip, or not, it's generally not EEG, but like the detector with the DBS can detect abnormal signals and then stimulate that portion of the brain.
Awesome. Okay. So the main reason we decided to tackle it and the main issue we're trying to solve here is that, like Mikhail briefly mentioned, it's fully invasive. And there are lots of additional surgery risks like brain bleeding, lots of side effects, swelling, stroke, infection, all these unnecessary risks that are associated and are now only able to be carried out with invasive. So the technology we're researching and the technology we're developing is using us beyond nanoparticles. So us beyond is an ultra small super paramagnetic iron oxide nanoparticle, which will then be guided by an external coil to be guided and like inflected by an external coil and magnetic field, then create the same effects and stimulate the nerves and the neural pathways deep inside the brain. So just to start it off, Anush, why don't you tell us a little bit about what happens once, how exactly the AV technology works, and what happens once the nanoparticles are in the brain?
Right. So once the nanoparticles and the magnet does the job of targeting the nanoparticles to the certain location in the brain, we are able to use basically electromagnetic coils that emit a certain frequency of radiation. And based on that frequency, we're able to oscillate these nanoparticles. And if you go back to high school physics, you could think of it as two magnets oscillating at a certain frequency to produce eddy current or an electrical field. And that's essentially what's happening in terms of the electromagnetic coil and the nanoparticle. And that electromagnet or the electrical field that is created is essentially similar to what happens naturally in the brain. So the brain thinks that it's actually an electrical field produced through just natural currents, and the impulse happens after that electrical field is created.
Awesome. All right. So I just realized I'll briefly mention the nanotechnology section, how that works, and then we'll follow up with Mikhail on the third aspect about how the imaging. So how the nanoparticle works is that first of all, we use iron oxide nanoparticles because iron oxide nanoparticles are a great agent for targeted drug delivery in general. And now we have to get them, this is the ultra small section. So the U as part of the us beyond is because in order to pass the blood-brain barrier, which is one of the strictest barriers in our human body, the nanoparticle needs to be a size under 20 nanometers. And now when it's at that size, we also get that property of super magnetism, that's to get the super paramagnetic. So once we have these fully designed super paramagnetic iron oxide nanoparticles, we then inject them into the bloodstream like a normal any injection would. And using its targeted drug delivery agent, it would automatically, and we bind it to the desired cell. So for example, if you're trying to target a specific disease like Alzheimer's, there's certain cells in the limbic system that get abnormal certain neurons, and those are specific cells we want to target. So before entering the body, we bind the nanoparticles to those cells, inject them into the body, travel through the bloodstream, past the blood-brain barrier, and physically bind to those cells. And once it do that, it also takes control of the ion channels, which is where it's able to actually stimulate, like Anush said. So that's sort of the first aspect. Then we would actually use the technology Anush said, the electromagnetic coil and the oscillation to stimulate it. And now after we do all that, let's follow up with Mikael on how the imaging side works, how we can actually get the results of our test.
Yeah. So basically, oftentimes when you have some form of fluid or some sort of structure within your body, and you want to use some form of medical imaging, you really need some form of contrasting agent. And so after hours of research, we came across a very revolutionary brain imaging technology known as magnetic particle imaging. And what it basically does, it applies static and dynamic magnetic fields to the supermagnetic iron oxide nanoparticles in order to get three-dimensional visualization of your brain, basically. And what sets MPIs apart from many of these other brain imaging technologies, such as MRIs, are its superb contrast, its very high temporal resolution, and its insane spatial resolution as well.
Awesome, that's great. So that's sort of the 3-in-1 approaches. Now, moving on, let's talk about the impacts. Let's talk about, like Mikael mentioned, some of the benefits. But Anush, I know I talked about some nanoparticle. On your side of things, what are some of the benefits of using these electromagnetic coils and nanoparticles as opposed to traditional deep electrodes in the brain? And then, let's start with that. You're muted.
Yeah. So the main benefit of the strategy is obviously that it's intravenously injected, so there's no invasive procedure required. But also there's a certain amount of resolution that we can gain on a nanoscale. Obviously, we're using USPIONs, ultra small, nanoparticles. So we could gain high resolution to exact locations of where the abnormal neurons are, modulating a certain frequency, and we could target that frequency using the electromagnetic coil. So that automatically adds a magnitude of accuracy onto our method. Another benefit would be that we're able to basically disperse this solution, which would usually be in like a liquid form based on our research, anywhere in our brain, and it could pass through the blood-brain barrier, and that's kind of like a Hans field of expertise. But we can easily place these particles in the limbic system and outside in the basically the neocortex as well, which gives us a overarching level of control over the brain. So that's the benefits.
For sure. So one thing that's sort of at an intersection between Anush, I think Anush might have skimmed over this, so I'm going to double down a little bit more. When the electromagnetic coil, how it actually works is you have each individual USPION, each individual nanoparticle is binded to a single neuron. So you have to have lots and lots and lots of these, but due to the fact that they're at this extremely small scale, that's not an issue. So how it works is the electromagnetic coil, this I'm going to go a little less technical so it might be easier to understand, is that it sends very low energy waves throughout the magnetic waves and electromagnetic waves through the entire brain, and then those low waves enough is enabled to oscillate the nanoparticle, which can then cause a stimulation. So the actual coil externally doesn't cause the actual stimulation. The nanoparticle being affected by it is what caused it. So now, Mikael, I know you touched on some of the benefits of MPI versus MRI, but what do you see as the future for MPI? When you're talking about it could be in this field as well as in this technology, even maybe if it could be applied to additional fields as well.
So in terms of like realistically, without like the nanoparticles in your body, or like MPIs would kind of be useless, and vice versa, right? So I would say that on top of just the brain side of like 3D imaging, you know, we could use MPIs to visualize other systems throughout your body, other organs, understand what's going on. For example, in your your respiratory system or something along those lines. So just, I would say, I definitely think that nanoparticles and MPIs are where it's at. It just comes down to like applying that to different parts of the body. And yeah.
Right. One quick follow-up question to that is that, do you think MPIs will reach the stage where they'd be able to completely replace MRIs? Because I know MRIs currently, there's something called magnetic resonance imaging. So if you could maybe touch a little bit on what that is quickly, and then talk about whether you think MPIs will actually get to that stage where they're able to be as commercialized as MRIs.
Yeah, so just to like touch on like how exactly MPIs work. So there's like 3 different stages. So there's like the signal generation and acquisition phase. There's like the spatial encoding and all, really like building that sort of image, and then the final reconstruction of the image. And like I said before, what really sets MPIs and what makes MPIs better than MRIs as of right now is really just spatial resolution, right? You're through those 3 steps, you're basically getting a three-dimensional visualization of your brain that is so powerful that you could even get quantitative data from it. So, which is like insane, right? So you're practically developing this whole image, and then from there you're getting quantitative data and like different signals, raw data from that image, rather than having something that's generally just for reading those signals. So I'm not sure if that answered your question, but I think
that great touch on MRIs as well. So just to sort of conclude, let's talk about a little bit of the future, and let's talk about quickly on the I was going to spend a little bit of time on the drawbacks of each technology, but let's talk more about the future, and then we can get into how we could overcome this as well. So starting off with you, Anush, is that using this electromagnetic coil and the nanoparticle what would be the future? What would be some of the more revolutionary aspects that can maybe be applied to other diseases or different stimulation techniques as well?
I think like outside of just general stimulation and like exhibiting or inhibiting proteins for basically diseases in the brain, there's also a positive benefit for enhancements in terms of the USPIONs. We could basically, when we oscillate the USPIONs, right, they produce heat. Well, because it's oscillating at such a high rate, and this heat, we could use it for eliminating tumors, like, you know, physical lesions, which is insane. And we could also use the heat to enhance certain certain neural pathways and mechanisms, and tissue conductivity, which induces stronger currents. So like, for example, for people with Alzheimer's, we're able to let's say like specifically the amygdala, which is the emotion center of the brain, we could, you know, increase tissue conductivity using the heat, which could possibly provide certain enhancements to their memory. And then I already touched on the physical lesions, right? So yeah.
Sure. That's incredible. So just to conclude, I'll talk about the future in my eyes for the nanoparticles. Is that my future is right now we're using nanoparticles primarily for the focus of just binding to the neuron. So in the future, as one of the more applications, we could design these multifunction nanoparticles, still at the same properties, all the USPIONs, they're ultra small, they're super paramagnetic, all of these things, But now we could, if Anush talking about different brain cancer stuff, we could be able to illuminate the tumors and even use it for targeted drug delivery. So we actually load the nanoparticle with the chemotherapy drug, and that we'd be able to, first of all, differentiate between healthy and unhealthy cells. So you wouldn't be getting any hair loss or any of those effects. But more than that, able to efficiently kill the tumor using the same nanoparticle. And then we could also use it as for MPI. So it'd be sort of an all-in-one package right now. Well, so that's sort of our technology. So just to conclude it once again, we were working on the 3-in-1 USPION assisted brain stimulation, which involves nanoparticle insertion, the actual stimulation through the electromagnetic coil, and the final magnetic particle imaging. All right, well, thank you so much for joining me, Anush and Mikael, and I'll catch you guys next time.
Thank you so much for having me, Sahan. It was truly an honor